Hello there, and welcome to this Biology Mind A-Level tutorial on signalling across cell membranes. So in the last lesson we learned about the functions of membranes. We looked at how cell surface membranes provide a barrier between the cell and its external environment, how movement is controlled of substances into the cell, and how cell communication occurs, including signalling and recognition. We also looked at how membranes around eukaryotic organelles compartmentalise the organelles within the cell, help increase surface area when they are folded, and form vesicles for substances to travel inside the cell. So today we have 3 key learning objectives. So number 1 is to overview of ligand-receptor cell signalling. Number 2 is to look at types of ligands, and number 3 is types of receptors. So let's crack on. So just to recap, to get us all on the same page, the cell surface membrane is essential for cells to communicate with each other. So how does this occur? For example, glycoproteins and glycolipids form receptors on the cell surface membrane. So remember, glycoproteins are just carbohydrates with proteins attached, and glycolipids are carbohydrates with lipids attached. They form receptors on the cell surface membrane, as we can see here, which are able to receive signals from neighbouring cells or from the environment. So for example, a ligand, which is just a substance that binds to a receptor, binds to our receptor, and this in turn is relayed to the inside of the cell to initiate specific cellular responses related to these messages. So communication across cells usually occurs via chemical signals. And these signals are known as ligands, as we said, and they are excreted by the sending cell into the extracellular space. And these ligands then go on to bind our receptors. These ligands are intended for a target cell, and the target cell is able to receive this signal because it has this receptor, which remember are glycoproteins, glycolipids, but at the end of the day, they're all receptors, and the ligands can bind to them, and specifically, a specific receptor binds to a specific ligand and stimulates a specific response within the cell. Now, when the ligand binds to this receptor, it triggers a cascade of reactions, and that ultimately leads to a change within the cell. Now, this can be small, such as activation of a gene, or it can be larger, such as inducing cell division. And bear in mind, a specific receptor is for a specific ligand. So most cells have many hundreds of receptors, thousands, millions of receptors, and various ligands bind to these receptors, which can trigger different intracellular reactions. Now, there are 2 types of ligands. Number 1, polar ligands, and number 2, hydrophobic ligands. Now, we've met this word before, hydrophobic, which is, remember, it's coming from a Greek origin. Hydro is water, phobic is from the Greek for hating, so water-hating substances. Now, this is relevant when we come on to talk about them, so we'll talk about that in a sec. But let's look at polar molecules first of all. So they only bind to the extracellular receptors on the cell surface membrane, as they cannot cross the phospholipid bilayer because they're polar, they have a charge. Now, examples include peptide hormones such as insulin. Some ligands are hydrophobic, so water-hating molecules. So remember, they're water-hating, they're going to want to burrow into our phospholipid bilayer and hide away from our water in our cytoplasm or in our extracellular fluid. So they pass through the cell membrane and bind to intracellular receptors, including steroid hormones such as testosterone and estrogen. So those are examples of our hydrophobic ligands. Now, we said there were 2 types of ligands, polar, number 1, and number 2, hydrophobic. So in the same way, there are 2 types of receptors. There's an extracellular receptor or intracellular receptors. So some receptors sit extracellularly, which means on the outside of the membrane, such as glycoproteins and glycolipids, and they're anchored into the cell membrane. So their ligand binding region, which we also call the domain, remains extracellularly, which means that the ligand does not need to pass through the plasma membrane in order to bind to the receptor, a bit like a polar ligand. Now, some extracellular receptors are coupled with another signalling protein that sits intracellularly. So notice that the ligand binding domain is on the outside, but the actual receptor itself is coupled with a signalling protein that sits on the inside. And how this works is, if we just go on to the next slide so we can have a zoom in, we have a ligand binding to our receptor. Now, this receptor we call a G protein coupled receptor. I'll just write that here. G protein coupled receptor. And all this means is it's a receptor that's coupled to a G protein, which you can see here. This is our G protein, this is our receptor. Now, when our ligand binds our receptor on the outside of the cell, we get a conformational change in our receptor. So it undergoes a conformational change, which just means that it changes its structure slightly. Now, what happens when it changes its structure is the G protein that it's coupled to. So you can see in this state, when the ligand is not bound, it's inactive. But when the ligand is bound, it's active. And that's because it swapped the GDP that it was bound to for the GTP. And this active G protein now uncouples to form a beta gamma part of it and an alpha part of it. And both are active in their own ways, and both can stimulate intracellular responses. So you can see here how an extracellular receptor, in which the ligand is binding to the outside of the cell, so to the outside of the receptor, is coupled with an intracellular signalling protein, leading to an intracellular response. Now, we looked at extracellular receptors, now let's look at intracellular receptors. These sit in the cytoplasm or the nucleus. And when their ligand enters the cell and binds to them, it causes the intracellular receptor to change shape and form a hormone receptor complex. So you can see that here. This is our steroid receptor, this is our steroid hormone, which, remember we said what sort of ligand was it? It was a hydrophobic ligand, so it's going to pass through our membrane, it's going to hide away from the water in our extracellular fluid, it's going to bind to our steroid receptor. Our steroid receptor is going to change shape and form a hormone receptor complex, and this complex is allowed to enter the nucleus because it's changed shape and causes a change in gene activity. So this is just a zoom in on that. You can see that we have our hormone receptor complex moving into the nucleus and stimulating a response. And if it's in the nucleus, what response will it be stimulating? Usually something to do with genes. So in this case, the complex is binding to sites and chromatin, activating mRNA transcription. Okay, so that's us done for today. Well done for following through to the end, and I'll see you for our next session.